Founder and producer, Professor Ahmed Mamdouh El-Sharif, Professor of Epidemiology and Infectious Disease, ex-head of Department of Veterinary Medicine, ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University, presents through this scientific veterinary channel. Technical veterinary learning. Jerusalem is our holy city. القدس لنا والأرض لنا. فلسطين حرة مستقلة من النهر إلى البحر. Malignant Cattrall Fever. Pathogenesis of Malignant Cattrall Fever disease. The incubation period in natural infection varies from 3 to 8 weeks. This is a proposed diagram for the pathogenesis of wide-range Malignant Cattrall Fever. A peripheral T-cell lymphoma induced by alpha-line herpes virus 1 infection. In the diagram, at the bottom of the picture, we illustrate the virus by this blue one that denote to alpha-line herpes virus 1 and the picture of regular and known DNA as cellular genome, and then transferase receptors and C8 lymphocytes and Alana with steak pink color, and then the alpha-line herpes virus episome. Infection to the cattle comes through nasal, saliva and contact, and then come out to enter the cell, then multiplication in the cell, especially lymphocytes, to make the different lesions, and then transfer to another animal. Alpha-line herpes virus 1 infect wide range of Malignant Cattrall Fever susceptible species via mucosal route, likely respiratory tract. Then host entry is regulated by both A7 and A8 envelope glycoprotein. Following such primary productive infection, CD8 plus T lymphocyte are infected by alpha-line herpes virus 1 through an unknown mechanism. In CD8 plus T lymphocytes, alpha-line herpes virus 1 establishes a latent infection, and a viral episomal genomes are maintained by ORF73 encoded ALANA in dividing lymphocytes. Subsequently, virus-induced cell proliferation ulceration leading to typical prevascular infiltrates in most of the vessels. Candidate specific gene of alpha-line herpes virus 1 are proposed at the end of this stage. Pathogenesis of this disease is the result of direct virus cell interactions, or perhaps immune-mediated responses that directed against infected cells. CD8 plus as a type of T lymphocytes are the predominant cells associated with the vascular lesions. Involvement of vascular adenivita accounts for the development of gross lesions, including epithelial erosions and keratoconjunctivitis, as present in head and eye fold. The lymph node enlargement is the result of atypical proliferation of sinusoidal cells. The cerebromeningeal changes, usually referred to as encephalitis, are in fact a form of vasculitis, which present in the nervous fold. There is commonly synovitis, especially involving Tibo-tarsus joint, and this is also associated with lymphoid vasculitis. Clinical
كل اللي
diagnosis.
سمع ولا
Mean
مسمعين؟
duration of
مسمعين
clinical
الثاني
time before
اللي معاه
death
سماعة.
is one outbreak
تعالي
was 6
اسمعي
days
وبعد كده مسكين.
with a range
بقيت البنات
of
زمايلي
1 to 26 days.
البنات كلهم سمعوا.
Malignant Cattrall Fever disease occurring in several forms. Almost clinical signs always present in cattle with malignant Cattrall fever are acute. Cases that suffering from fever, corneal edema as present in this picture, oculonasal lesion as present also in this picture, and oral lesions. Malignant Cattrall fever forms, all graduations are present per acute form, subclinical form, head and eye form, alimentary tract form, common head and eye form, and mild form. Initial clinical signs accompanied by fever with depression, diarrhea, dyspnea, persistent high fever from the beginning until to the end life of the animal, and inappetence. Fate of all forms. Acute form ended by sudden death. Common head and eye form. Majority of cattle cases progress through different stages. Intestinal form, initial head and eye form but die of severe diarrhea. This picture indicated lesions that occur on the tongue and oculonasal discharge that clear here. Mild form, inoculated animals will be recovered by means that this is an experimental infection. Head and eye form. This is a clear picture of this form. This form beginning with reddened eyelids, then blepharospasm and uveitis, then bilateral corneal opacity as in this picture, with presence of crusty muscle nerves, erosions on the tongue. Head and eye form also shown, erosions on the tongue, erosions on the buccal mucosa as appear here in this picture, and the yellow arrow denoted to it. Nasal discharges, salivation, lymphoadenopathy. Bovine malignant Cattrall fever showing joints, superficial lymph nodes are swelling, horns, hoof coverings are sloughed, nervous signs, incoordination in gait after about 4 days, head pressing at the end of the stage, nystagmus, and hyperesthesia at the end of nervous form. Malignant Cattrall fever disease showing horns and rarely the hoof may be shed as appear in this picture, and the black arrow shows it. Persistence of the fever from the beginning up to the end. Acute encephalitis which cause the nervous manifestation. Dies reported 7 to 10 days later on from the beginning of the first sign. Illness lasts for 3 to 7 days and rarely up to 14 days. Nervous signs that reported during malignant Cattrall fever: weakness in one leg, then incoordination, then adheminated appearance with muscular tremor and nystagmus. Also affected animal as appear in this picture will suffering from head pushing, paralysis, convulsions in the final stages, trismus, with superficial lymph nodes are often visibly by means that enlargement, gross hematuria. Mild form of malignant Cattrall fever that reported in water buffalo, bison, and some species of deers. It is observed in natural outbreaks in these animals. With transient fever, mild erosions appear on the oral and nasal mucosa. Recovery reported with recordance. Maybe this mild form is changed into chronic form with long convalescence stage. The animal will have persistent bilateral ocular leukomata. Animals with bare acute form showing a short course of the disease 1 to 3 days ended by death with the following signs: a high fever, dyspnea, acute gastroenteritis, fulminating disease. This is common in deer and in Egyptian cattle. This picture showing eruption of the pupil at the end of the head and eye form. Alimentary tract form resembles the head and eye form with marked diarrhea, minor eye changes, conjunctivitis rather than ophthalmia encountered in outbreak form. Alimentary tract form encountered in outbreak and cattle in large dairy herds in dry lots, in direct contact with sheep, reported. Attempted and farmed deer will be infected. Fulminating disease in deer by mean begun and ended in severe manner. Mucopurulent discharge as appeared in this picture and muzzle crusting with presence of adherent fibrinonecrotic material that partially clouds the nasal orifices. Post-mortem diagnosis. This picture showing the erosion that appeared on the tongue of the animals during head and eye form and alimentary form. Lesions in the mouth, nasal cavities and pharynx vary from minor degrees of hemorrhages and erythema through extensive severe inflammation to dissecated ulcers. These lesions may be shallow and almost impenetrable or deeper and covered by cheesy diphtheric deposits. These lesions are very clear. Erosions of the tips of the cheek papillae, especially at commissures, is common, and it is annoying at that part because have to be differentiated from cattle plague and mucosal diseases. Erosions of the tongue appear clearly at the left picture and on soft hard palate appear clearly on right picture. Hard palate ulceration with presence of diphtheric membranes appear at this picture. Ulcerative and necrotic pharyngitis and laryngitis. Necrotic areas appear as diphtheric membrane in the larynx are often present. Erosions at the tips of the cheek papillae and other signs in this case have to be differentiated from mucosal disease, blue tongue disease, and cattle plague. A clear picture of tongue erosions. There are longitudinal, shallow erosions are present in the esophagus. Similar but more extensive lesions occur in the abomasum. Catarrhal enteritis of moderate degree and the swelling and ulceration of Peyer's patches are constant. All of them are reported during the alimentary tract form. This is a clear picture of esophageal lesions. Abomasal mucosal surface showing necrosuppurative ulceration with yellow-green exudate at the tip of the yellow arrow. Necrotic areas in the omasal epithelium. Multiple erosions were noted in the intestinal epithelium. In case of malignant catarrhal fever disease, all lymph nodes are swollen, odometous, and often hemorrhages appear. The gross ocular lesions as described clinically. Corneal opacity and mucosal petechiae are clear in this picture. Longitudinal section through inguinal lymph nodes, and you can note the loss of normal tissue structure of the organ and necrotic appearance of these lymph nodes. Irregularly distributed white to reddish foci of a few millimeters in diameter deform the surface of the kidney. The liver is swollen and severe hemorrhages may be visible in the urinary bladder. A picture of kidneys of an animal dead from malignant catarrhal fever showing raised tiny white foci on the cortex according to the arrows. Highly enlarged lymph nodes. This is number 1 compared to normal one in number 2. Urinary bladder mucosa are hyperemic and odometous. Keep on the track by following the scientific channel Technical Veterinary Learning for more accurate scientific discuss and clearance of all infectious diseases belonging to animals all over the world. Thank you for subscribing.